Hi, everyone. Welcome. We'll give it a few more seconds for everybody to join, and then we can start. Okay, we're on. Good morning, everybody, and welcome to today's high throughput collaboration webinar. I'm Sharon. I'm the VP of Operations at CodeOcean, and I'm delighted that you all joined us today. Before we get started, a few housekeeping items. First, the session is being recorded, and we'll send it to you after the event. Second, you are more than welcome to submit your questions throughout the webinar using the questions pane. Should be, I think, on the right-hand side for most of you. We will collect these and address as many as we can during the Q&A session. Please don't be shy. I'm thrilled to introduce our speakers for the event: Yair Benita, Head of Science Operations at CytoReason, and Simon Adar, Founder and CEO of CodeOcean. Please take it away.
Thanks, Sharon. Hi, everyone. So I'm Simon Adar. I'm the co-founder and CEO of CodeOcean. My background is in signal processing, image processing, spectroscopy, hyperspectral imaging. And I started CodeOcean out of Cornell University. And our mission is really to empower researchers such as yourselves with easy-to-use computational tools so that your work is really efficient, reproducible, collaborative, with the ultimate goal to achieve high-quality scientific discoveries. And Yair and I, we know each other for many years now, and we every time that I speak with him, I learn something new. So we thought that it's going to be nice to share some of the knowledge with the rest of you. Yair, maybe you can start with just saying a few words about yourself before we get started.
Sure. Thanks, Simon. Hi, everybody. Thank you, CodeOcean, for the opportunity to speak. I'm Yair Benita. I lead science operations at CytoReason. My background is in pharmacology. That was my bachelor's of science. Later, I did a PhD in bioinformatics in the Netherlands, in Utrecht University, working on gene finding in the genome in the early 2000s. I did my postdoc at Mass General Hospital in Boston, where I was for the first time exposed to patients, and that inspired me to move to pharma companies. I worked for Merck for 5 years as a computational scientist, and in 2014 came back to Israel to lead the computational group at Computational. And since 2017, I'm with CytoReason. I lead, as I said, the science operations, and basically I'm responsible for all our customer projects. CytoReason, briefly, is a company that works on human disease data. We believe in collecting all the human data, integrating it, and making it into a disease model which can be analyzed and queried. And we believe this is the path forward to increase the success rate in drug discovery.
Awesome. So just before we start, what will you learn from this webinar? We're going to talk about how to design a productive team. Specifically, we're talking about the Compbio team, as well as how the Compbio team integrates with the rest of the company. Key processes and best practices that enable collaboration, and the technology stack to support the daily activities. And we're going to finish up with do's and don'ts, personal perspective, Yair's personal perspective. It's sort of a rapid fire type of questions, and then we're going to open up for Q&A for the audience. So, yeah, you know, I was looking at 2020, and it looks like biotech is exploding, and I'm asking myself, why now? Like, why IPOs are 3x in 2020 compared with 2019? And you know, the amounts of funding that biotechs are getting in the marketplace is growing so rapidly. Why now? What's happening?
Yeah, that's a fantastic question. So I'm going to try to answer it by going through my personal perspective and growth, which I think is indicative of what happened in computational biology in the past 2 decades. Basically, when I started early 2000, I was working on cells, mostly in vitro cells. Then later started to move to data generated from animal models as we tried to study more and more diseases. And today what we see is that most of the data comes from human. We moved from studying cells to studying humans in a very short amount of time. And that's actually extremely exciting because the human is the real thing. We're suddenly are able to understand disease biology better. It's no longer models. Then if you look at what happened on the data side, you know, early in 2000, when I started working, we generated most of the data in the lab. Some of it was online, but then it was quite limited. And then we started generating more and more data through robotics. We could make, like at Merck even in 2009, we generated millions of data points in 3 months in a single experiment. And today we're actually getting massive amounts of data from patients. We took all these tools, all the technology, and started applying it to biopsies. Then on the technology side, what we saw is initially, at least, we studied a single cell. That's the next build. We studied a single cell by PCR. That's actually a single gene by PCR. That's how the genome was actually put together. And at some point, we were able to look at all genes, initially through microarrays, later through sequencing. And now we're even at a technology, we have a technology where we can look at a single cell from a patient. Imagine collecting thousands of cells from a patient and profiling each one individually. That provides tremendous value. And finally, on the analytics side, we started with, you know, using even Excel to analyze data from small experiments. And then the data became too big. We needed to analyze it using the computational tools to really write code. And now the data is so massive and you need so many skills that you really need a team of analysts to be able to handle it. So really what all of that comes together, if I bring it back to your question, what is happening now? If you look at the outer circle, we have a really high resolution technology. Generating the data is not so expensive. We can do this from patients and we have the analytical capabilities. All of that means you can really peek into what's happening in humans in disease. And on top of that, there's an area which is not my expertise, but I will mention there's really unbelievable biological technologies to target different proteins and processes in humans like CAR-Ts or CRISPR to edit DNA, which are making a huge impact on where you can interfere or intervene with a specific disease. I want to stress that point of human research on the next slide. I made that slide a while ago, but actually it brings a really cool point just to show you guys how much data, just to show everybody how much data we're generating in clinical trials now. So this pie chart actually shows cancer indications. It was published in 2006, about 800 trials of combination therapy in immuno-oncology alone, so relatively small therapeutic area. And if you're assuming, if every dot here is a trial, if you're assuming just 50 patients per trial, you're looking at data from 40,000 patients. Now, the cost of that data is massive. A patient in oncology in a clinical trial costs about $1 million. So that's $40 billion invested in testing all these drugs. And it's clear that most of them will fail. They will never make it to market. But actually, the massive amount of data that were generated, the companies are generating for R&D purposes, DNA, RNA from biopsies, is tremendous and brings a huge amount of value.
So it sounds like the opportunity here, if I'm hearing correctly, is that the precision medicine that people are talking about for decades now is now becoming a reality. Is that what's happening? Is that the opportunity?
Yes, I think the opportunity is definitely that we have a way to make the decisions way more data-driven. Initially, when people looked at it, they said, this is not hypothesis-driven. This is just carpet bombing of the field with a lot of money. But actually, what we're starting to do today, let's say you tested the combination in melanoma, by analyzing quite a lot of this data, which is, by the way, mostly private, you can actually say, well, what your drug does in human is not what you anticipated exactly. You should move into lung cancer, where it fits the mechanism a little bit better. And your decision become more data-driven. So there's an opportunity to be smarter about how we test it, because as you can see, the cost is quite massive. And the cost is to test the drug. It's not to generate the data. Generating the data is sort of a byproduct which can feed into a much better decision-making process.
So all of this, was not really available a decade ago, and it is now. So what is the new role of the Compbio team? How do you see that now?
Yeah, so I think the role is definitely evolving. Let's start with the role as at least I experience it in most pharma companies that we work with. I mean, I'm sure many of you online can think about your bioinformatics group. So typically it's made of individuals with different skill sets. Some of them come from the genetics field, some are chemists, some have experience with protein networks, the microbiome, they have different disease backgrounds. So they're a collection of individuals, and typically what I see at least is that there's no software engineering skills within the Compbio team, and typically also no biologist or clinician, people who don't write code but are expert in the disease area. And what really happens right now is that mostly individuals are assigned to a specific project, and they support it. They help the biologists answer some questions, and they provide the analytical perspective. But the thing is, with this model, you actually rely quite a lot on the skills of one individual. It's really difficult to come up with a standard way to do an analysis. For example, let's say if you give each one on your team a task of, let's say, deriving a biomarker from clinical trial data, you'll be probably relying a lot on the skills of the individuals, and you will get very different results based on the different people who did it. In addition, at least from my experience, and I'm a computational biologist mostly, the code that we write is mostly done for one analysis, and we're not good as computational biologists in writing code that is reusable. I can say for myself, probably 99% of the code I wrote was used just one time for an analysis and was not used in many projects. So that role, so I think that structure is good for a role where the Compbio team provides statistical analysis services to most biologists. But I think that role is changing now. Mm-hmm. And given all the data that I just showed, the human data, the Compbio team is now expected to come up with hypotheses on how a certain disease, what's the mechanism of action of a certain disease, what is the biomarker for a drug, which target should the company follow on a specific disease of interest. So very big questions for pharmaceutical companies that are based on a lot of data, and it's no longer a task for a small group of individuals with statistical background. It's a task for a much bigger team.
And that's what goes back to what you said about the change in the last 2 decades from doing an experiment. The biologist goes and does the experiment on single cells, and on the contrary, right now we have massive amounts of data, and now we need to think about what's the hypothesis from a data perspective. And you and I, we had a discussion about methodologies. It sounds like we're now trying to bring together a lot of data sources, multimodal data sources, and trying to understand the biological system, which is extremely hard. So how do we do that? How do we build, different capabilities? And we talked about maybe taking software development methodologies into the Compbio team. Can you tell us a little bit about that?
Yes, absolutely. So I've been thinking about that problem a lot. How do you build a team that can, within a few months, tackle a very big problem? That's like, what's the disease mechanism of psoriasis or arthritis or something like that, just from data. And I realized that we need not only to figure out what kind of skill sets to put in such a team so that they're able to execute it, but we also need methodologies. And as you said, we're in our field in the transition from a single person running a project to a team. And actually, software development went exactly through this process. If we're thinking back to the 1980s, where a single person was able to write an operating system, of course, today it's inconceivable that one person will write an operating system. But that field actually generated not only many different roles on a team, but also development methodologies, right? So in terms of roles, we have a front end, a back end, we have the designers, we have architects, so we have many different types of people. And in terms of methodologies, Agile, or what Spotify came up with, the squads, there are many different methodologies of how to work, and sprints and all these methodologies that were invented. And of course, that field became very successful. So I think in the analytical world, we're also at the position, probably in the first transition into a team, and that's what we experimented with. I at least experimented with in the last few years to see what could work for us.
Yeah, it sounds like almost inevitable, like the amounts of data, the complexity, and the different types of skills. It sounds like you even need more skills on the Compbio team than the traditional software development. So sounds like there's sort of a gap in the lag between these two worlds, and that needs to be shortened in terms of methodologies. So
right.
we're going to pause here for a poll number one. We have another one coming up to ask the audience, who are you collaborators on? Who are you collaborating on specific research projects? And you can select, different multiple answers. So we're going to give a few seconds for everyone to bring up all the different options. And Sharon will help us with.
I'm a second before you all, and it looks good. It's very exciting. I can see the results coming in. I'll give it a few more seconds. This is changing live. Okay, I think we're good to share.
All right.
Let's do this.
Okay, well. Hey, what do you think? Is this surprising? You see, scientists, biologists is 90%, which is the majority here.
Yes, I think it nicely confirms, at least for me, the model where most computational scientists actually provide services to biologists. So it's not surprising that I say provide services. I don't mean it in a bad way, but usually the biologists are conducting the experiments, and they need, because the data is on a large scale, they need assistance. And with clinician, the collaboration is lower. So in that sense, it's not very surprising. Yeah. I think it makes a lot of sense. I'm surprised by the bioinformatician and computational biologist. I thought it's a trick question and they should be equal. Not sure what happened there.
Yeah, maybe those needs to be sort of combined together. I'm also, it's very interesting to see that external partners many of the people here are working with an external partner, and maybe this shows also the evolution in biotech, biopharma, and companies like CytoReason, right? You work primarily with external partners. You work with biopharma, and it shows how that changed also.
Very interesting.
Yeah, very interesting. So that takes us to the next thing. I think is a great segue for the concept of the new ideal in the Compbio team structure.
Yes, so I wanted to share here the model that works for us at CytoReason, which I think we optimized and works really nicely. So we have a team that is sort of an independent team that can execute on a relatively big question from pharma, like what is the target, or give us a target for a specific disease, which is in a data-driven manner, right? So we don't go to read the literature, we need to do it from data. And there's quite a lot of data out there. So a team is typically composed of computational biologists, a biologist, a clinician, and a software engineer. So the computational biologist and the biologist are always full-time on that project, but the clinician and software engineer can be part-time. The challenge here is that these are very different skill sets, and these people don't really speak the same language. And when you tell a biologist the word a model, he thinks about a mouse model, or she. And when you're thinking about and you pose the same question to a computational person, he would think about a statistical model, right? So they don't speak the same language, and it's actually very challenging. But what happens when they work together and they're actually full-time focused on that one project, they start to communicate, and the biologist would say, I don't really understand what this data, or you need to help me, and the computational person will help the biologist, and they start building this language together. And by working side by side, they actually come up with very cool ways to tackle the problem, and also they start to understand each other's worlds, right? The clinicians start to understand a little bit more the molecular data and the statistical methods, and that cycle evolves.
And when you say that they work together, do they physically sit next to each other? I know today everybody's working from home, but traditionally saying before COVID.
Absolutely. At CytoReason, they work together most of the hours of the day. It's really a ping pong, a back and forth all the time. We have a specific questions. We look at the data doesn't make sense, so let's try something else. So it's this back and forth all the time, with a weekly meeting to sort of say, here are the main things we found this week, and this is what we want to do next week, and bring everybody in, collect the opinions. And basically, the Compbio team is now conducting research much faster than you would in a lab, right? Imagine if you have a question and you want to ask it in a lab setting. Now the experiment takes 6 months, especially if it's in animal models. Here, you can ask a question and get the answer in 20 minutes. So you're progressing very, very quickly towards an hypothesis, asking questions for all sorts of directions. So all these people are involved. It's actually really fascinating to see.
And it also, reminds me what you mentioned about the methodologies, the software development methodologies, and Spotify. You mentioned the squads. So basically, that's the Compbio squad.
Exactly. When we got started, I thought it was going to be difficult to manage multiple teams and basically keep expanding. So the idea was to bring in people with strong expertise in their field, put them together, and make sure they're capable to execute on their own mostly. What myself and the other managers mostly do is to keep them focused on the problem, provide the client perspective, challenge them to get to a specific answer. But they do most of the work. They really they have all the skill sets to do it fast with the software engineering, to think about the data with the computational biologist, understand the disease, the molecular, the cells. They have everything they need, and they come up with very cool ideas, which we then want to reuse, right? So that's also one of the points. That's, by the way, the role of the software engineer. let's make sure this is reusable.
So that's the squad concept, and how does that fit with the rest of the company?
Yeah, so that's as we started expanding, we realized we need a central platform. We don't need to recreate or start a project from scratch every time. So what we started to do is to we built a relatively big R&D team, which contains both software engineering and algo developers. They build the platform and they deal with the with both the infrastructure and let's say more difficult or they should be testing on the algorithm side a little bit better, and basically packaging everything so that the scientific teams can use it. We don't want a team that now goes into a biomarker project to start exploring how to generate a biomarker, right? So that needs to be packaged. And if one team had to do it, then the engineering team would wrap this into a capsule that works. So I do want to mention the concept of Capsule, which worked nicely for us. First, we work in a cloud setting, and CodeOcean is part of our platform. The capsules are a great way to package the environment, the data, and the code. So a scientific team that actually built a specific project or made a breakthrough can provide that capsule back to the engineering team to make it part of the platform. And that's how we started to build standards, make sure the code is reproducible. And as our tools matured, we are now starting to actually provide access to customers because they're stable enough, they're usable. Our biologists and clinicians like it. So we think we can train also external customers on those tools and make sure they benefit from it.
So it sounds like you're both working on specific projects that your customers are looking for answers. As part of that, you are building blocks, and you mentioned biomarkers. It's one, but aggregating different types of building blocks from different projects, and gradually you have a platform of a library of so many different building blocks that you can bring to the next project. And then also, the next level for you is actually to expose that to your customers as a platform.
Exactly. Many of these problems have shared components. Like if you're thinking, for example, about dividing a disease to subtypes, maybe something that works in atopic dermatitis does not work in RA, right? And although we did it once in one disease, we had to optimize it for another disease. But they have a lot of shared components, and we make sure that at least the shared components work well. And as we capture more and more information within a disease, we also now package the components per disease so that it's easier, because our field actually works on by disease. There are pharma companies are divided to therapeutic areas.
Yeah. And you mentioned the compute capsule as a concept for collaboration. Can you tell us a little bit about how that works in sort of standardizing sort of the collaboration here between the different roles and the teams?
Yes, absolutely. So I remember when we talked, a few years ago, I told you that early in my PhD, I spent most of my time compiling code of other people so that it runs on my Mac. It was very difficult, and it was also very frustrating. The concept of the capsule is that everything you need is actually packaged for you in an environment that works. You have all the libraries you need, you have all the code you need, all the packages, everything is there. And the capsule is like a template for your analysis. If you think about even like a PowerPoint template, the capsule is a template for a biomarker, for disease stratification, for target ID. And the way we work is that the capsule is initially designed by our lead scientist, who makes sure everything works, everything is there. And it's a template that different groups, science groups can use. Once they use it, it's like a forking in Git, right? You have everything you need. Now you apply your specific project, and sometimes you can improve it, or you solve a specific problem that was not solved. You can share it with other teams. You can say, hey, I actually in this project, I solved it. Why don't you look how I did it? You can give it to others. But more importantly, we can give it back to the lead scientist to see, look, in this, because the team is an independent team, they can improve the method and give it back, and that the lead scientist will ensure that our template gets improved all the time and is updated. So that actually way of sharing makes sure that we improve the platform itself.
Mm-hmm. Yeah, and it sounds like it's a sort of an iterative process that is happening here. And obviously the concept of the compute capsule can be implemented, in different ways by people in the audience, not necessarily, using CodeOcean, but the concept of having something that captures all what you need in one environment that has the code, data, computing environment, and you are basically able to track back in terms of versioning of the entire project.
Absolutely. This was, by the way, a big consideration as we started to work with companies. We wanted everything we do to be reproducible. Every plot we generate, every piece of code, be able to track everything. And actually, the compute capsule provided that for us as well.
Yeah. So this brings us to the next poll, the second poll here. When we were thinking about the projects, every project, every computational project has a result. So asking the audience, what is the preferred desired collaboration deliverable of the project? And there are different possibilities here. And Sharon can update us on the progress of results coming in.
Yeah, again, I think these results are actually surprising. I'd love to hear kind of Yair's thoughts, but I think it's better than I expected.
Oh, you're building tension. Now I'm curious.
We'll give it a few more seconds in case anybody else wants to put in an answer. Okay.
All right.
Let's do it.
Wow.
Okay.
Very encouraging.
Yeah.
First, I'm really happy to see the interactive analysis scoring at the top with 72%. This is definitely the way to go. I'm really happy to see it. So I guess notebooks is a bit surprising because then it's a result that is usually you would give to another computational person and not a biologist. Data sets, and PowerPoint's at 50%. That's interesting to see.
Yeah, but I think it really brings the point here that interactive analysis is considered something very, very valuable. Actually, we would be happy to hear from people. Maybe people can put in the chat, and Sharon can sort of share that with us. But taking this to the next slide.
I still see the poll. Oh, okay.
Yeah.
I would actually mention that we got a response from Peter that he actually uses none of the above.
Oh.
Yeah.
Interesting.
It'd be interesting to see what kind of other file types are there.
Yes. So I was very excited to see the interactive visualizations. I think it's super important because this is what we need to communicate with biologists, right? Biologists typically don't write code, and their input is super critical. And what we really want is to give them the option to fill the data, to play with the data themselves. So I recognize this early on in my career. I can say that initially it was very difficult to provide interactive visualization. In 2008, during my postdoc, I actually built a static HTML website, which is still online more than 12 years later. but it was very difficult. I invested a lot of effort in it, and it's very difficult to update. Never touched it again. But today, actually, what we do is we have 2 ways to share visualizations. One, in CodeOcean, it's actually very easy, like a Shiny app. You can actually, in just 1 or 2 minutes, build an app panel that runs R or Python code and generates the plot, and it's interactive, and people can look at any type of visualizations that you code without, let's say, they can write the gene name or the disease and play with it. The other alternative is to provide an interactive viewer, such as Tableau or Spotfire, which is typically used in pharma companies. We actually provide our biologists with a Tableau interface that is linked to data that is generated on the CodeOcean platform. So we have the capsule actually also contains the output of the code, and Tableau can connect to this. And then the biologists can build their own graphs once they understand once they understand the data. They can start to put together these visualizations and very quickly start to ask questions on their own. They don't need a bioinformatician anymore. You don't need to peek over the shoulder of someone and say, why don't you make this plot for me? You can do it, and then the research accelerates tremendously. So I'm very happy to see that people do it. I'm not sure how long you spend building these interactive visualizations. I hope not too long.
Yeah, but it sounds like there's also a mixture of things: the static reports, the presentations that people really need, maybe for decision making, but a lot of people are thinking about leveraging the work to provide some kind of a tool at the end.
Exactly.
And that's great. That's great to see.
I think biologists also probably demand it today. They're not content with just a slide. When you show somebody a slide, immediately has another question. Oh, but what happens when you do this, and what happens when you do that? So it needs to be interactive.
Yeah. We want to give enough time for questions, so we want to wrap things up and sort of narrow the lessons learned that you had in the last 2 decades into something more concise. What can you tell the audience in terms of the most important things from your side?
Yeah, the most important things that I've learned on putting together a team. So I think what really worked for us is to put together at least 2 people, bioinformatician and a biologist, and give them a relatively big problem, such come up with a drug target for a disease, or stratify a disease to subtypes, or find an indication for this drug, right? What would be the indication? A relatively big problem, and then limit their time to, let's say, 4 months maximum, not more than that, and they can only use data, right? They can't do any experiment. They can use any data that the company has or that is out there in the public domain. Now, initially, this is a very hard problem, especially for biologists who would think about what experiments to conduct. But there are no experiments, no exceptions. And the key here is to allow them to run this project full time. It can't be a side project, because then they would go to do other things and they will not progress enough. They need to work on it every day, all day, and they'll start working together. And the biologist will say, your data doesn't make sense. And the bioinformatician will come back and say, I don't know, this is the data, and they'll try to figure out how to explain it. Maybe there's a bug, maybe not, but if there's not a bug, they'll come up with all sorts of questions to follow up on and to progress. And then I guess the third point is, at the end, we should not really let them just provide a presentation. That doesn't work. We need them to give us a tool that will allow us to fill the data. And the reason is that kind of transparency provides trust. And that trust is critical for people who make decisions, because the goal of this computational biology effort is to really make better decisions as we develop drugs. So what I suggest is, towards the end, force them to sit in front of somebody who's actually making those decisions. There's somebody in a pharma, typically a VP or SVP, who we'll have to make a decision of whether a drug progresses to a phase 3, or whether they should go to a specific indication, or whether they should kill the drug altogether. Each of those decisions is worth hundreds of millions of dollars to the company. Now, the key question is, can you come up with a strong enough analysis to convince somebody like that to make a decision? And I, as the one who's basically running our science operations, that's the role I typically try to take. And I force the team to come up with one answer. If it's a drug target, give me the one. Because scientists will never give you the one. They will say, well, if you want this, I can, that's what you should do. If you want something else, that's what you should do. There are many options. We don't understand enough the biology. And that's all clear. We don't understand enough the biology. There are gaps in the data, but still we need to make a decision. What's the decision? Yeah. And when you force them to make that decision and present it, it's actually, they come up with very cool data and analytics.
And in terms of limiting their time, what's your interaction with them during those 4 months? Do you do it daily, weekly, monthly? Do you sort of let them go and come back to me after 4 months and let's see what happened?
No, so we don't do that. Typically, we meet on a weekly basis to see where they are, what they're doing, and really my role here is to play the client perspective, right? To look at what they're doing and ask, okay, how is this helping me to make a decision? If I need to choose which indication to go into, is this even helping me? And keeping them focused on the problem. That's the main thing. Focus on the problem. Is the analysis you're going to do next, will it help us make the decision, right? So I try to focus them. That's what I mostly do. And I typically would meet with them on a weekly basis.
That's great. So we're coming to the last part, which is going to be a rapid fire questions. I'm going to ask you a question one by one, and you can answer as quickly as you can. Sure. But before that, we are going to start collecting questions while we're doing the rapid fire questions. So for the audience, please feel free to write questions. Sharon is going to collect those, and we're going to do it just after the rapid fire questions. So the first one is Python or R. There are two communities. Which one?
Yeah, definitely both for me. So most of our platform is in R, but Python has its strength, so definitely both.
Okay. There are different ways where people are doing their analysis: laptop, on-prem clusters, or cloud. Which one?
Yeah, definitely cloud computing. That's no doubt. Both for the speed, transferring huge amounts of data from clients. Also, all the protocols are there, HIPAA compliance. Everything we needed is in the cloud. Really, no reason to choose anything else.
And what would you say for people who are talking about that the on-prem is actually much cheaper in terms of the hardware?
Well, I can't imagine building something on-prem for us. I mean, the amount of compute we're using, storage, all the security that is needed, everything around it seems to me like a huge operation. It's for a company like us, it's not in our core, so it makes much more sense to just buy it from the big vendors.
In terms of the way that people are calling themselves in terms of the title, we're hearing bioinformaticians and computational biologists, and we actually in this presentation we use those interchangeably. Bioinformatician or computational biologist?
I have no idea. For me, for me they're interchangeable, but maybe I'm just not up to date. I don't know. I'll be happy to hear additional thoughts.
That's actually going to be interesting. If people have opinions on whether we should call this bioinformaticians or computational biologists, or how much distinction is actually between those two groups. Next question is build or buy a computational platform.
So my personal thinking is if it's not something that is a core competence for the company, just buy it. It's not worth it to develop it and maintaining it.
In terms of scientific publications, Nature, AACR, or Science Magazine?
For me, AACR, mostly for the meetings. it's a great, I mean, I grew up in the oncology field, so AACR is a warm spot in my heart.
East Coast or West Coast?
Oh, definitely East, because of the time difference. Can have meetings before 9 p.m.
And you also, you spend a lot of time in the Boston area, right?
Right, exactly.
ggplot2 or Plotly?
Plotly. I love Plotly. The visualization is awesome. It's really nice.
Tableau or Spotfire?
Tableau, no doubt, it runs on the Mac. Spotfire, as far as I know, as far as I can recall, at least from a few years ago, doesn't. Maybe they fixed it by now.
In terms of pipelines, what's your recommendation? What are you using? Bioinformatic pipelines.
Yeah, we have our lead scientist who's super talented and is our pipeline engine. So we were not using any commercial platform.
Last question. If you had to choose a secret power, as a Compbio secret power, what would that be?
I would say bug hunting, to be able to quickly find all the issues with the code that are so painful to sometimes find.
That's awesome. All right. We're going to go now to any type of questions that are coming from the audience. Maybe Sharon can help with that.
Sure, and please keep the comments coming and the questions coming. We have great engagement. It's great to see. One comment and question that I thought was interesting to discuss, we got from Thomas, and it goes this way. When interrogating the role of a single drug target in a complex disease process, it's important to empirically validate the relevance of hypotheses and insights provided by the analysis of very large data sets. And Yair, we talked about it so much. And biologists are skeptical when computational analysis yields unexpected results. What considerations should be made for determining how often working teams should pivot between bench research and computational work? And is there an ideal strategy for having both research and computational modalities guide each other during the lifetime of a project? That's it, an easy question.
A lot of content in this question, many challenges from Thomas. Yes, this is, of course, I have my share of pain trying to convince biologists of a certain analysis. It's definitely challenging, but this is the reason. So I think the key here is that if a computational scientist runs the analysis on his own and then presents the end result, people can be skeptical, right? They see it for the first time. Let's say you give a presentation. It doesn't make so much sense to them. It's really difficult to absorb. Now, that's the reason I said that it needs to be a team where the biologists work side by side with a computational person. And when you start doing that, what happens is that throughout the process, they see everything that you're doing, right? And then they are part of reaching the conclusion. So they won't be skeptical. The reason we have a biologist on our team is exactly because of this tension. Giving computational people, telling computational people to go and now present their analysis to a customer is more likely to result in that issue, that in skepticism. But actually what I want is for them to convince the biologists sitting next to them every day, having lunch with them. And once they convince a biologist inside the company, we manage that tension internally. We send the biologist to present to the customer. If the biologist is convinced, he will know how to talk to the clients. And this is how we manage that tension. Make sure that the biologists are part of your process. Be super transparent. Give them access to the data. Tell them that's the reason we have an interactive tool, right? Say, look, come and see the data for yourself. You don't believe it? What don't you believe? Start to work with them, right? That's the way to convince them. At the end of the day, as I said, we have to convince a decision maker who doesn't work with us. And imagine a VP who needs to make a decision on which indication to go to. How do you convince them? You have to be fully transparent, build your story very tightly. It's difficult. It's definitely not easy, but that's the reason you need the team, a team with the clinician, with the biologist, and with the computational scientist. I hope that helps, Thomas. What was the second part of the question?
I think you kind of touched on it, but the second part was, is there an ideal strategy for having both research and computational modalities guide each other during the lifetime of a project?
Yes. Well, I just want to make one more point then. The first thing I did when I came to CytoReason was hire biologists because there were only computational scientists here. I think you need full-time biologists, people who don't write code on the Compbio team. It's not typical. I don't see that a lot. I sometimes see it because we do work with many pharma companies. I do see it, and actually it's really cool that it's there. But most of the biologists that people collaborate with are actually bench scientists. They work in the lab, or maybe they're directors, but they're in charge of a wet lab operation. I would add biologists to the Compbio team. That's, I think, is super important. Make sure they're there all the time.
Great. Another question that just came in a bit of a different direction is what best practices do you recommend when sharing with an external partner? And I guess that builds off on kind of what we've seen in the survey.
Mm-hmm. Yeah. So I think the same principles apply, except with external partners, and we struggle with this all the time. You can't be fully transparent because you're risking the IP that you're developing. Let's say if we came up with this great algorithm and we think it's a competitive advantage, we can't really give the code to a collaborator to, let's say, the real purpose of sharing is to build confidence, right? If we want them to accept the results, we want to share everything. We want to share the data, we want to share the code, we want to share the algorithms. We have to share everything so they can look at it and say, wow, that works. We believe this analysis. But when you can't share, it's more difficult to believe the analysis. So I would say be as transparent as possible. We always, we don't share the algorithms, but we actually share the data and the results in an interactive way. They can visualize it on our platform. They can play with it. We give them ways to benchmark. Oh, you don't believe this? Here's a way to test it in a way that is intuitive for them. Look at cell to gene correlation. Look at anchors in biology that everybody accepts as general truth, okay? To build that confidence. So the trick is to really build confidence in result. It's slightly more difficult with customers, but it's definitely doable.
Is there any iteration, Yair, just a follow-up question on that, where you're completing a project that goes to the customer, and then there are more questions or a follow-up project according to the results that were generated?
Yes, absolutely. There's a tension all the time. Let's say the bioinformaticians from the customer side will always ask for more. How did you do this? Type of question, right? Show me how you did it. And we try to provide as many details as possible without compromising our technology, but there's always that. The biologist will keep asking us biology types of questions where this analysis doesn't make sense to us. How do you explain it? And it's a constant iteration. We have monthly meetings with them and we keep pushing through exactly as we do internally, but on maybe bigger intervals.
I'm taking you to a whole different direction. And another question that came up is, when do you decide to version analysis work? I think that's a wide one.
Oh, so good one. Within a project, once we started working on a question, we freeze all the analysis. Like we even if, let's say, some of the technologies got updated during the project, we don't use them because we don't want to change the result. It's too tricky. So within a specific project, we don't allow for versioning. But after a project, definitely yes. Unless sometimes we have to, if we figure out that a certain analysis type doesn't work for that disease or that data or something, and we have to do it, then we get input. We pull people from the R&D team to help fix it. But other than that, we don't touch versioning within a project, only between projects.
Okay, we're going to do last question for today. We're jumping around a little bit between topics here. Yair, you've mentioned that you're kind of building, you're preaching to build teams differently than they are today, than they are built today. What pushback did you experience through the process, and where do you anticipate kind of challenges?
Yeah, so there could be some pushback in terms of hiring people who are not computational to the computational team, right? And I think it's also challenging for biologists to join the computational team and become a bit more data scientists and basically leave the lab. In my experience, biologists have a really hard time in the first month, but then let's say after 3 months, they if they push through 3 months, then they're okay and they start to be real data scientists. But other than that, I don't think there's a lot of pushback. It's a bit difficult to find the right skill sets for a team, but I didn't, at least we didn't have any pushback internally.
Okay, just wrapping things up, I wanted to thank you so much, Yair, for coming in and sharing all this knowledge and your lessons learned with us and the audience. For everyone that if you have any more questions or both for Yair or to me, feel free to reach out. We are going to follow up with a recording and some other materials that you might find useful for after this webinar. And again, we're happy to hear your feedback and the types of topics that are interesting for everyone, and we can follow up with. Thanks everyone. Thanks, Yair.
Thank you, Simon. That was a great opportunity. Thank you, and happy to follow up with everybody who wants to discuss how to build teams. Please reach out.
That's great. Thanks everyone.
Thank you.